So good morning, everyone. Today I will talk about our approaches to control Campylobacter in chickens. So Campylobacter is one of the most common causes of bacterial foodborne illness in human. It's considered a commensal bacteria in the chicken gut, and infected chickens serve as a reservoir and shed bacteria throughout their lifespan. Contaminated poultry products are considered the main source of infection in humans. And 500 bacteria can make humans sick. Symptoms are ranged from mild to bloody diarrhea, abdominal pain, vomiting, and in severe cases, gastrointestinal perforation and Guillain-Barré syndrome. Therefore, to reduce the risk of human campylobacteriosis, it is essential to reduce the bacterial load on poultry products. Various intervention strategies have been applied to reduce colonization rates, including biosecurity measures, sanitation in slaughter plants, dietary manipulation, antimicrobial alternatives, genetic selection. However, these strategies have been only partially effective in controlling Campylobacter burden in poultry flocks. So vaccination is considered a key part of controlling infection. However, no commercial Campylobacter vaccines are currently licensed for use in chickens. In addition to that, the limited protective efficacy of the current existing vaccines signifies the need for more efficacious vaccination strategies. A large body of experimental evidence suggests that TLR ligands can be exploited as adjuvants within vaccine formulation or as a standalone antimicrobial agent. TLR ligands comprising microbial components and their synthetic analogues. Previous studies have shown that in ovo or parenteral administration of CpG-ODN, which is the synthetic analog of bacterial DNA, can protect chickens against enteric infections such as E. coli and Salmonella typhimurium. We have recently screened various TLR ligands for their ability to stimulate chickens' cecal tonsils mononuclear cells, and we found that CpG-ODN has an overall greater immune response induced an overall greater immune responses in chicken cecal tonsils mononuclear cells in comparison to LPS and PAM3 CSCK4. This has led to suggestions that CpG-ODN can be exploited as a potent adjuvant, especially in the context of enteric infection. So we hypothesize that oral delivery of CpG-ODN will lead to induction of intestinal immune responses, which may lead to breakdown of commensal relationship of chickens and Campylobacter. However, oral delivery of CpG-ODN remains a challenge due to the short half-life of the DNA and potential degradation by gastric juice and digestive enzymes. So efforts have been made to overcome these shortcomings and increase intracellular availability of CpG-ODN by formulation in protective delivery system such as PLGA nanoparticles. So these nanoparticles actually facilitate target delivery of CpG-ODN to antigen-presenting cells, enhances vaccine immunogenicity, facilitates actually the target delivery, enhances vaccine immunogenicity, and protects the TLR ligands from degradation by digestive enzymes, extends the duration of immune system stimulation. Our results revealed that CpG-ODN reduced the Campylobacter count by 23 folds in a chicken intestine, which means approximately 19-fold reduction of public health risk in humans. As we can appreciate here, encapsulation of CpG-ODN reduces Campylobacter burden by 46 folds in chickens and approximately 29 folds reduction in public health risk. We also use CG-Joni lysate as a multi-antigen TLR-rich vaccine, and we found that oral administration of this lysate reduces Campylobacter burden in chickens by 114 folds, which means a reduction of incidence of infection in humans by 32 folds. A greater reduction was achieved following a concomitant delivery of PLGA CpG-ODN and CG-Joni lysate, as we can appreciate here. So in conclusion, Oral administration of CpG-ODN alone significantly reduced intestinal colonization of chickens with C. jejuni. PLGA encapsulation with the nanoparticles enhances the ability of CpG-ODN to reduce C. jejuni load in chicken Zika. And oral administration of the combination of PLGA encapsulated CpG-ODN and C. jejuni lysate can serve as a candidate vaccine for control of Campylobacter in broiler chickens. At the end, I would like to express my sincere thanks to my advisor, Dr. Shaheen Sharif, Dr. Doug Hodgins, Leah Reed for her technical support, lab members, isolation staff, and the granting agency. I would be happy to entertain your questions.